very much for the opportunity to speak with you today. I thank the ASS, and it's great to have my friend and colleague moderating this session. And it's nothing like being told the punchline to spoil a good movie. And I hope that I'm able to fill in some additional information for you today on the work that we've done, looking at utilizing a very specific resource, the platform of microgravity, to develop potential therapeutics for on-Earth application. So just in terms of an overview, what we are interested in is the development of therapeutics for infectious disease. We want to specifically use microgravity exposure to uncover targets for the development of these therapeutics. And that's a little bit different from understanding all of the mechanisms, because for some of the bacteria that I'll talk about today, mechanisms of infectivity are known. But what happens is this will change, as you've heard, in the microgravity environment. The missions that we've been fortunate enough to fly are part of the ISS National Lab Pathfinder vaccine missions, and that's kind of a mouthful for really utilizing space to validate this as a platform for potential commercial development. I will say that the work that I'm going to talk about today, and all of our work with the Pathfinder mission payloads, has been done on shuttle. We've specifically chosen bacteria that have small reproductive times, and this is kind of for this approach is sort of the low-hanging fruit. That is, understand the platform that you have and validate the use of the time you're given in that platform for going on farther, as Julie mentioned, for studies that require longer periods of time in microgravity, which will become very good candidates for development, research and development in National Laboratory on ISS. And very kindly, NASA has provided us the flight opportunities to accomplish our goals. If it hadn't been for some very strong advocacy at headquarters through Mark Uran's office and Bill Gerstenmaier, we wouldn't have had the opportunity to have a manifest on now 10 nearly consecutive shuttle flights. And with 133, when it launches, will be our 11th payload. And I want to, this is great that I have this platform because I get to thank friends and colleagues like Julie Robinson and Mary Beth and Dean for their help, because it's really hard when you go up as a soft pack and you're not part of the original program, and you have to find space to be able to do this study when things break and things can't go up on time on this flight and have to be deferred to another flight, and these are the people that make it all happen. We're very appreciative. Well, we all know that infectious disease is a tremendous problem. It kills more people in the world than many of the other chronic illnesses combined. In our country alone, the cost due to diagnosis and treatment of infectious disease is in excess of $100 billion. So this is a very costly problem, and I just read a report released from the UK last week that the biopharma market, which is what it's now being called, last year had a value of $78 billion and is expected and predicted based on modeling to increase 15% per year. So a huge amount of money is being put into the development of therapeutics for this very costly problem. And this is a ripe area to utilize unique conditions to make an impact on trying to reduce this economic burden. We need vaccines and therapeutics, and although the title of this is vaccines, but really we're talking about vaccines in the context of the larger umbrella of therapeutics for disease, to stop the spread of disease and to address the increase in antibiotic resistance that's happening in the population of microbes at large. We all know we go to the airport and we sanitize our hands, and we have our special soap that we wash our clothes in, and now more and more use is focused on antimicrobial agents that reduce these organisms in our daily living. And what that is also doing is creating super duper organisms that are smart and adapted and are able to circumvent these countermeasures that we're trying to enforce on them. Our approach then was basically to take advantage of these missions that are left, that were presented to us on the shuttle flights and now going into the end of the program, to use this as a foundation to validate new gene targets that will be uncovered by exposure to the microgravity environment. And the group was formed, and our work was initially started with the launch of STS-123 in March of 2008. And that was really, it was the first time that we asked the question, can we approach this platform from the perspective of commercial R&D to take what's been known about an organism and to capitalize that for potential therapeutic development. It requires a lot of partners. We have a lot of academic partners. The principal investigator of this work is Dr. Timothy Hammond, who's a professor at Duke University. He is also on the staff of the Duke VA Research Hospital. He has also now been just accepted into a position at VA headquarters. So he's got a really big job and big opportunity in the VA system, as well as his academic activities, part of which is dedicated to this Pathfinder work. We also have colleagues across the globe that help us by providing knockout clones that we've used in our work. We've worked with people at University of Zurich and the Max Planck. We have a lot of colleagues at Duke. I'm at Baylor, and so we have a very strong academic basis. The work is funded, though, by industry, and this is through a small spin-out privately held venture called Astrogenetics, which is held under a larger parent aerospace company, Astrotech. And the very important partner here is government, without which we would not be able to do these studies, with NASA providing the flight opportunities, and also we work through a CRADA with the VA hospital and system, so that we're able to use two arms of government to support our work. And that becomes important when we talk about submission of intellectual property. So I think we already have established, I apologize, I didn't get a chance to take some of these slides out, but because you already know why space. Space causes changes in bacteria. The changes may be associated with growth, they may be associated with changes to antibiotic resistance, pathogenicity, or virulence. So again, the bottom line strategy is to take advantage of this new knowledge so we can treat infectious disease. And in the beginning, our question was, if we can understand the microgravity-induced changes in gene expression, and if we can identify those changes, can we use this knowledge to create a new vaccine? So for the standard tried and true vaccine, which is an attenuated vaccine in biotech lingo, it has to be strong enough to induce an adequate immune response in the host, but it cannot be too strong to induce sickness in the host, and it also has to induce recall, or the ability to have the immune response generated when the system of the host sees that bacteria again. So in terms of scientific design, we leveraged a lot of ground-based work. Our initial studies were done with Salmonella. Tim Hammond was part of the team that did the fundamental work in Salmonella that showed when you take the organisms on shuttle and let them grow under microgravity conditions, you bring them back and you put them into mice, that you see increased killing of the mice, correlating with enhanced virulence of the organisms. We then leveraged a series of ground-based assays to optimize host-microbe interactions, because we wanted to be able to develop a system that we could ask questions rapidly within the microgravity environment. And we took advantage of a C. elegans system that had been developed by one of our collaborators at Duke, Alejandro Abele, that allows us to have a very easy-to-work-with host that mimics a lot of the interactions between the bacteria and that host that we see in humans. We then leveraged existing flight hardware. You saw some of this presented yesterday from Louis Stodiak with Bioserve, who has so much experience in biotech and biotech plant and microbial experiments, and he has been incredible and has been our integration and hardware partner for all of our studies. And we developed an in-flight host pathogen assay. And as I mentioned, the little organism is C. elegans. It's a little tiny worm that lives in the soil. And the bottom line is C. elegans eats bacteria, which is really nifty because that's what we need to happen. And for Salmonella, it's a gut organism, and we wanted to use this to validate this model as an effective host pathogen model so we could look at some new targets. So what did we do? Let me see if this is a microscope shot of C. elegans, and it has some animation with it. You see the little worms wiggling, and it's really fun. But anyway, we're not going to get to see that today. That's our crew. But basically, we use Lewis's gaps, and these are the individual tubes within each gap. There's 8 of these tubes that go within one of these canisters, and within each of these individual little compartments, we will have bacteria, then we will have the organisms, and then we will compress those 2 on orbit by the crew, which are trained by Lewis's staff. And the experiment is carried out, and then a final push will inject the organisms that have been incubating with the host into a fixative, and the experiment is stopped. If the bacteria infect the worm, they eat the worm, eat the bacteria, they get into the gut, and if they kill the worm, what happens is there's no more worms there, and the bacteria are allowed to grow, and that's reported as increased optical density. If the bacteria do not infect and kill the host, then they simply get digested. The worms eat up the bacteria, and you do not see the increase in optical density. So it's kind of a quick and dirty yes/no test in the beginning. Of course, we do dissections, and we look at worm-host interactions and adherence and actual infection within the gut lumen. And this is some typical data. This is actually one of our first experiments. And this shows you what happens when we took out specific genes that we postulated to be associated with this increased virulence response, because it's by subtraction. If the gene that you take out is associated with that response, then you should see a reduced OD or reduced virulence effect. And in fact, this is one of our first target genes that we identified, oddly enough, in our first mission. And so, being the good scientists we are, we couldn't believe it and repeated this several times. And in fact, we have another gene that looks just like this. So we actually have 2 really good targets that we've identified in Salmonella. Now, what I want to talk about too is it's not all just about virulence, and I want to talk about host pathogen interaction, because for Salmonella, as well as for most organisms, you have to have an attachment or a binding of the bacteria to the host cells or tissue, and then you have an invasion. So what we do is we actually use 2 kinds of worms, a wild-type worm and then an altered worm that's deficient in a known binding protein for the bacteria that we're working with. And that kind of gives us an insight into is the virulence response associated with both a binding as well as an invasion process, or can we uncouple those and study them